Everyone. I'm Richard Owen. I'm president and the CEO of the American Peanut Council. And I have to tell you, I've been looking forward to today's Growing Together webinar for a few months from the time when we were able to secure our speaker today. I'm very, very pleased to introduce Dr. Charles Simpson, who's going to share with us really his lifetime of research on the peanut species. He's been responsible for discovering, collecting, and maintaining the peanut wild species collection in Stephenville, Texas. And the stories that he's going to share today start all the way back with the jungles in South America, with 28 different expeditions searching for both cultivated and wild peanut cultivars. Altogether, he and his team have collected over 1,800 accessions of wild peanuts, including identifying 75 new species. Dr. Simpson's been a peanut breeder and germplasm specialist at Texas A&M since 1967. He earned his bachelor's, master's, and PhD from Texas A&M as well. He semi-retired in January 2003, and in talking to his colleagues, semi-retired still means coming into the office almost every day and dedicating his work in life to continuing what he's going to share with us today in his presentation. Also pleased to recognize that he and his colleagues maintain one of the largest peanut wild species collections in the world. So with that introduction, I'm going to turn it over to Dr. Simpson, who's going to take us through his journey in identifying the wild peanut species in different places. I would ask that if you would please put your microphone on mute. He's going to give his presentation, and then we've saved time at the end for questions from the audience, and I'm sure there'll be lots of questions. So Dr. Simpson, with that, it's over to you.
Okay, let's do the screen share. And I think we're ready to go. Yes. Good morning. And thank you to President Owen and the American Peanut Council for the opportunity to talk about my favorite topic, peanut germplasm. Now, there are two ways to look at the title of my presentation, the germplasm collection itself or collecting the germplasm. And I want you to think of it in both ways with me for this presentation. Now, I've spent the last 53 years working with the collection as it grew, and I've spent 43 of those years in actual collection work of the germplasm. Now, I don't want there to be any confusion about this. I've been a member of a team of collectors. Yes, a leader and a co-leader and a member, but the collection work has been a team activity, with several of us playing key roles at different times in different places. Now, the collection we maintain here at Texas A&M AgriLife and the other collections in the United States and other countries, such as Brazil and Argentina and ICRSAT, will be the very lifeblood of the peanut industry in the future. With the genomic information now available, and the APC helped fund a lot of that information, and the new technologies that are being developed, these plants are where the genes will come from to meet our needs for variety improvement for many years to come. Therefore, it is of utmost importance that the germplasm collections be maintained, because many of the original populations no longer exist in nature. And there are still population to be collected. I'm sure of this, and Dr. Valls and Dr. Sejo would agree with that. With that said, I'm going to move on into the presentation, and I think, yes, there we go. Now, the peanut genus Arachis originated an estimated 5 million years ago, and that figure is debatable. The cultivated peanut was developed about 3,000 to 5,000 years ago. Again, debatable, but that's my assessment. Now, the peanut genus probably originated in this region here in Southwest Brazil, Northeast Paraguay, sometime after a mid-tertiary uplift of that old Brazilian shield. This area here, the old Brazilian shield, the uplift occurred sometime in the mid-tertiary, about 30 million years ago. And the genus probably originated in that eroded shield, and the remains of that shield still exist today in Northeast Paraguay, Southwest Brazil. Those eroded remains. I never did get to the top of one of those, but I bet there's some interesting things there. But the reasoning there is, as Dr. Gregory and Krapovikas found, that the most primitive, morphologically primitive species of the genus still exist in that region. Now, this is a wild peanut. 3 leaflets. They don't look like peanut leaves to most people, but does have a peanut flower and fruits and pegs. This is Arachis guaranitica. Now, the unique thing about peanuts, and it's not totally unique, there are a few other species that produce their fruit underground, but the peanut produces fruit underground, and that presents some issues with how did it get distributed over the large area that it covers in South America. And this shows the size of the fruit of wild peanuts compared to the cultivated, and a hybrid between the wild and the cultivated. And this picture shows this peg here. These are pegs, not roots. This is a Arachis paraguayensis, section erectoides. The pegs grow horizontally, and this is a mechanism that has evolved to get the seed away from the mother plant for distribution. And so if that peg is about 1 meter long, it could move, say in 4 million years, it could move about 4,000 kilometers. And running water has played a significant role in that. And we see this from the air. We see the movement of the water during the rainy season, and these loops and turns where a peanut seed could be deposited in these areas. And we actually find peanuts in those. This in the Rio Grande River in Bolivia. These sand dunes, we actually collected in some of these sand dunes, collected wild peanuts growing in these areas that had come downstream from above. Here in Argentina, Arachis duranensis, the A genome donor to the cultivated peanut, there is a duranensis plant growing in this driftwood. Now, the peanut genus, the wild peanuts are found in this region here, from the mouth of the Amazon to the Northwest Bolivia, Argentina, Northwest Argentina, down to Uruguay, up the Brazilian coast and back. This area is where we found the wild peanut growing. We've also collected cultivated, and I'll get to that in a moment. In the mid-1960s, this sort of thing started showing up in the forest in South America. They would come in and bulldoze the trees down and put them in, or push them in rows and start burning, and then start growing crops in the usually upland rice. Here, this is Dr. Valls, and I'll talk more about him later, but this was the population of Arachis marginata. They built a road, they cleared the land, they had planted upland rice, and we found this population of Arachis marginata growing in the rice field, in the road, and in the uncleared forest. And this is a shot of some of the road building when we could get there before they paved it, before it was too far gone. We could find the populations growing in the edges of these roadways. In Brazil, in southern Brazil, in 78, they were growing sugarcane for the alcohol production to run their cars. In this area here, they pushed down the forest and to plant grasses to graze their cattle. and soybean fields prepared like this, uh, if a population of wild peanuts, whether it be a small population or a large population, by the time they put it to the plow and the herbicides, the populations will be gone. And here in Northwest Argentina, again, the Duranensis, with the original site, they built a oil refinery on that area. We could still find it growing in the between the materials, but once they put it to asphalt and concrete, it'd be gone. Now, these three men were the real pioneers of the collection of material. Dr. W.C. Gregory at NC State, Jose Pietrelli from Argentina, and Antonio Krapovicas from Argentina. They collected in 1959 a 6-month trip, and again in 60 and 61. But a lot of the materials that they were able to collect did not survive as germplasm, only as herbarium specimens. So Dr. Gregory was really important person in international circles, and he and others helped the formation of the International Board for Plant Genetic Resources that was formed as part of CIGAR, an international agency, and the FAO, a part of the United Nations in Rome. Their objective was to preserve peanut, not just peanut, but all plant germplasm. Then in 75, Dr. Gregory and Krapovicas submitted a 3-year proposal to collect both wild and cultivated material in these countries. These were the areas where they proposed to collect, and these, of course, these would be in the Andes just for cultivated, but this was for wild species. And the first expedition was in 76, the December. I was not able to go on that one, but the second expedition was my first in 77, Northwest Argentina, and we split the team in Bolivia to three members to the west into the Andes to go collect cultivated, and in east central Bolivia to collect wild peanuts. 4 of us went there. And early on in 77, 78, 79, when we did what we call airstrip hopping, we would land in on these strips. This would be the only contact that these people had in Northwest Bolivia with the outside world, that and a boat. And so we land on those strips, and we do a lot of walking. You see Dr. Gregory and Scanini here. We did a lot of walking and a lot of collecting. Now, in 1980, Dr. Gregory was forced to retire, and so he turned his part of the leadership over to me. And at that time, I assumed the responsibility of the bearer of the funds and became a leader of the materials. Also in 1980, Dr. Jose Valls was hired as the peanut curator and also forage grass curator at Embrapa San Argine in Brazil. And in 1981, he and I made our first expedition to Western Mato Grosso. This is Emil Grip, a driver for us. And these are some of the roads that we traveled in the Western Mato Grosso. It took us about 2 weeks to go less than 200 miles, but we collected about 5 new species in that trip. This is just a shot to show an Arachistendosperma that originally was evolved in the central part of Brazil, but was carried to many places in Brazil by the native people. The peanut has evolved wild species have evolved in some really interesting places. This is in the Caatinga forest in northeast Brazil, a very dry area, about the kind of rainfall that El Paso gets. And these slides from Dr. Valls show how dry it can get. The materials flower with the rain grew and flowered and fruit, made seed, but then really dry situation. And then from that into the Pantanal, which is a large area, swampy area. During the wet season, these ponds fill up, And the wild peanuts, as in the dry season, as it dry up, the wild peanuts seed deposited grow along the margins. This was the objective. One of the objectives of the first trip in 1976 was to recollect Arachis diegoi at one of these lakes, Laguna Gaiba. From that then westward, as the genus evolved into more advanced materials, Arachis betizikoi, here we've collected at about 2,000 meters elevation, Arachis betizikoi. So a wide variation of materials. Now, there are some really beautiful bridges in South America, in Argentina and Brazil and other countries, but this is not one of them. This is one that we crossed successfully. Dr. Valls drove and I took the picture, but we traveled some very interesting places. Now, when we come upon a population like this, where it's flowering like this, Arachis decora, this again from Dr. Valls, these are easy to spot, but this is early in the season, early in the wet season, and you likely not find any mature seed. So you either have to take live plants if you're collecting, or come back at a later time to collect mature seed. Here we were collecting from a Mamadi Indian tribe in western Mato Grosso. They were showing us how they were growing a wild peanut for food in their slash and burn field. This is a slash and burn field. In Paraguay, we encountered this group of Guarani tribe that we showed them a herbarium specimen of a wild peanut, and they knew the plant. They walked us about 5 kilometers to the west into those sand dunes I showed you earlier, and they knew the little armadillos were digging something, but they didn't realize what. And after they see my little collecting screen here, after we collected some seed, they wanted to know if it was, I told them it was a wild peanut. They wanted to know if it's good to eat. So I shelled one and ate it. And while they really started buzzing in their native language, that here was a source of food that they didn't realize that they actually had. On that expedition, Dr. David Williams was co-leader of that. And this shows the GPS that we were introducing at that time to give more exact locations. And from that point on, we developed some uses of the GPS much more accurate in our locations. Now, let me back up one. Our charge from IBPGR was to collect the materials, seed or plants, and leave a sample in the country where we collected with someone that knew some about the materials, if we could find someone, so that we would leave material with the country of collection. But the charge was to bring material to the United States and multiply it and then distribute it to the national collection here in the US at Griffin, Georgia, and to the international collection at ICRISAT in Hyderabad, India. And we did that as best we could. But then in, I think it was 1993, a conference of most all South American countries, and they voted to suspend the approvals for outsiders to take germplasm out of their country without agreements to share royalties. And the USDA was invited to this meeting, but they had no authority to sign such an agreement. And in 1997, the pact was finally ratified by all the countries except Paraguay. And so we were still able to get bring materials out of Paraguay. And in 2002 and 2007, we returned to Paraguay and collected. But then after 2007, it also stopped in Paraguay. But in Paraguay, we collected this wild species that we named Arachis flugi. And this species is going to be very important to us in transferring genes out of the wild peanuts into the cultivated. Now, one of the things that I was assigned from the very beginning was to collect the nodules from the live plants and put them in these desiccator vials, bring them back to the United States, give them to Dr. Elkin at NC State University. He would isolate the material, the bacteria, and put them on glass beads. And as far as I know, they're still available in that form. Many of the U.S. The inoculum have some of the strains that we collected in their makeup. Pardon me, let me get me a swallow of water. This is Aurelio Scanini. He's a Paraguayan botanist who worked at in Argentina with Dr. Krapovickas. He and Dr. Krapovickas made herbarium specimens when we were together. They made herbarium specimens of almost everything. And then in 2004, again with Dr. Valls, a more advanced GPS system. And after I retired in 2003, I continued to work with Dr. Valls in collecting. And my objective was to help get materials collected and get them into the Brazilian collection, so that eventually we hope that someday we can get things worked out between the governments so that we can still get the materials from the other countries as well as Brazil. But the GPS has been very helpful in locating materials. Now, talk a little bit about the cultivated materials. From the high Andes, we would go into areas and take some real risk in crossing rivers and things like that. Here we crossed the river and talked to this peanut farmer. She had two varieties, one that grew on top of the mountain and one down in the river valley in the dry season when the water subsided. And we collected both of those varieties. Now we have collected about 5,500 land-raised cultivated lines and about 1,800 wild species lines, 75 new species or more. And these are the areas where we have collected from Ecuador to Brazil and Uruguay and Argentina and Bolivia. And in 1994, Gregory and Krapovickas published the monograph, did the taxonomic work and described 69 species, including the cultivated peanut. And I was fortunate enough to be able to contribute to 7 of those descriptions. So I was a co-author on 7 of them and listed 69 species. Now, this is an awful busy slide, so I'll not spend a lot of time, but these are the 9 taxonomic sections that they assigned. And the more primitive at the bottom, intermediates, and in the middle and the top are the more advanced materials. And the cultivated peanut belongs to the Arachis section. They have, the cultivated peanut has 40 chromosomes, and there are 2 species of the Arachis section that have 40. The other species is Monticula, which the Hypogea, the cultivated peanut, was derived from, and 31 other species that have 20. All of those cross with the cultivated peanut readily. And then the rhizomatous type, there are 3 species that have 40 chromosomes, 1 to 20, and the others mostly have 20 or 18. And I'll not spend any more time. I just want to point a characteristic of each section. Here, the cultivated section with the taproot, the rhizomatous section with rhizomes, the Erectoides section with the tuberous root system, and the tuberous root system on the extra nervosi, this Arachis marginata, the Coleorhiza, again, the slide from Dr. Valls showing the 4 different flower colors, the Triseminata section, 3 seed on a peg, and of course the one I showed you earlier, Triseminale, the 3 leaflets. Dr. Valls and I described 11 more new species in 2005. Dr. Valls described another species in 2013. Dr. Valls and I described another new species in 2017. And Dr. Sergio Altrez and I described another species. Actually, it's just now accepted into 2020. Dr. Viles and Dr. Sajo have more new species yet to be described. Now, in the preservation of the germplasm, most all of the cultivated germplasm lines can be stored for a long period of time with the seed that are produced. I have some cultivated material that have been stored for more than 40 years. Some of the seeds still germinate. About 65% of the wild species produce enough seed to be stored for long periods of time. However, there are some of those species, the 15 of the Erectoides section, that they cannot be stored for long periods of time on a dependable basis. So we have to maintain live plants. Therefore, with these and the 35% that do not produce seed, they must be preserved as live plants. So to have a complete collection of wild peanuts, you must have a good seed supply of materials, but also a large live plant collection. Just a few examples of materials that express the different morphological features of the wild peanut, Arachis lineafolia with the small leaflets, Batisacoa with the large leaflets, early leaf spot reaction. We find resistance to that. Late leaf spot, we find resistance to that. We've combined those into materials that releasing varieties from that. The susceptible and resistant to Melodicon arenaria. Here are species that is highly susceptible to Melodicon arenaria root knot nematode, one that is highly resistant. We transferred that into the cultivated peanut and released this variety and other varieties that have been released from that. We transferred leaf rust resistance from this species, Arachis cardenacei. And there will be other things that come from Arachis cardenacei, but this is only one of the species that resistant to 5 pests so far, just one species. And what is to come from the other 81? These wild species will be the lifeblood, as stated before, of the peanut breeding for many years to come. And also from the cultivated materials, the resistance to tomato spotted wilt that we developed Tamarin 96 out from came from a plant that was collected in northwest Brazil. And I just wanted to point this out, that on the cultivated materials, when we bring them to the United States, I'd grow them 2 generations in the greenhouse just to make sure that there was not a problem with having introduced some disease or insect or whatever. And then after 2 generations, I would take them to the field. This is just to show some of the variability in the seed coat colors and size of the cultivated peanut. Now, our efforts in our project, we are using the genomic information and the Raman technology. We are working to identify and utilize genes for drought resistance and quality traits. And this is what we are spending a good bit of time on. The Raman technology is a new way to analyze plant tissue in a non-destructive analysis. It's a laser-based technology that utilizes a 1-second laser blast that gives a graphic reading of the content of what is being tested. And after a catalog is built, a laser reading can identify a seed that are resistant or susceptible to an identified trait. As an example, nematode resistance or protein content such as lysine. This slide I borrowed from Dr. John Kaysen, showing the traditional gene transfer integration. It take about 20 years. With using some of the modern technology and transfer of root knot nematode resistance took us 13 years. Marker scented back crossing, 9 to 10 years. And the Raman activity, maybe 8 or 9 years. And cisgenics and gene editing, that sort of information, maybe we can cut it down to 5 to 7 years for variety development. And with screening and trait identification, phase 1, movement of traits to elite cultivars, phase 2, and agronomic testing under field conditions, phase 3, to develop new varieties. Now, we have one of the most complete wild peanut germplasm collections in the United States. Many of these can no longer be recollected in their native environment. And we have 76 of the 83 described species. And we are upgrading our facilities. And we have new personnel, and the one I want to mention specifically is Dr. John Kaysen, the new peanut breeder here at this location. And we have, they're forming an endowment that is being established to support the use of the collection and maintaining the collection here for a long time in the future. And with that, I think that I have reached the end of my presentation, and I'll be glad to try to answer any questions that we have.
Great, wonderful, Dr. Simpson. That was very interesting. You only scratched the surface of some of the highlights of what you've done over the years in identifying those species and bringing them forward for the industry. And I think the industry has a big debt of gratitude to give to you for all the advances that have led to the current cultivar we know as peanuts. The questions have started to come in, and the first question is actually similar to one that I was going to ask you. So you had a lot of adventures in South America and Brazil in particular over the years. What would you identify as some of the most memorable adventures? What are just a few of the highlights of the most memorable adventures you had during your research?
Well, it's awfully hard to describe how much of a high you can get when you're down on your knees, digging in the soil, sifting with a little screen or just by hand, getting these small seed, and you realize that, hey, this is something new. There's not a high anywhere in the world for me that compares to that. You maybe you haven't eaten for a couple of days, and maybe you've been stranded, like we became stranded in Paraguay for 10 days. Fortunately, the Paraguayan army was coming by and rescued us. But there's no high that can compare to that.
Great, thank you. Only a few people, I'm sure, even in the research community, can share that kind of experiences that you had like that. Another question came in. What are some of the challenges that you faced in preserving and creating a duplicate of the collection that you discovered for the future? Some of the challenges during the collection and preservation.
Oh boy. Well, there have been issues in being able to transfer material to the international location in India. I have had some real issues over the years in transferring material, and that was one of the charges, that we would multiply the seed of the seed producers and get them in the hands of the international agency, ICRASAT, in Hyderabad, India. And we we finally did some things that were not according to the way ICRSAT wanted them done, but I won't say they were illegal. It's just that they were not following the rules that had been laid out. I'll put it that way. But we did get a lot of the material into the ICRSAT program, and it was really frustrating that I could not send things directly from my location, and that was really frustrating for me. I had to send them to another location and have them send the material from their location. And that was one of the real challenges. I don't know if that answers your question. The person that asked the question, if they want more detail about something more specific, I'll be glad to try.
No, that was good. I think that answered the question. And if not, then if the person could ask a follow-up question. So another question came in asking, would it be possible for you to comment on how much the distribution of A. durensis has been altered by ancient human activity?
How?
The A and then durensis, D-U-R-A-N.
Durensis?
Yes.
What
has been how much the distribution has been altered by ancient human activity?
Oh, of Durensis, I don't really think that the Durensis was one of those that was highly involved in the by the hunters and gatherers. There are other species that were, in my opinion, but Durensis does not fit into that category, in my opinion. There are some archaeological evidence of wild peanuts being grown in northwest Peru, but Durensis is not included in that group.
Okay. I think that answers the question. Another question came in about where your collection is located. And the question is, how much of your collection is at the USDA facility in Griffin, Georgia? But I would ask a broader question. So what different locations are your collections scattered to?
Well, the charge that we had was to get it into the national collection in Griffin, Georgia. And originally the materials were transferred. Well, we made the first trip in 76, and then 77, 78, 79. And it took a few years to get the material multiplied. But then I don't remember exactly the first year that I transferred material to Griffin, but over the years, they had materials, but with issues in funding and other things, it seems like with the and I don't want to stomp too many toes here, but it seems like those of us in germplasm preservation, when budgets need to be cut, one of the first things that gets cut is these long-term things that require a lot of funding, maintaining a germplasm collection. And that happened in Georgia in Griffin. And because of that, there were a lot of materials that got lost. And we've been in the process the last 4 years working with funding from the National Peanut Board and the Peanut Research Foundation to try to replenish many of those that had been transferred before, to make sure that they are at least in the national collection, and they can take it from there to distribute as required. The distribution of materials it's a time-consuming thing, and I've had requests here over the years, and of people who send me before email and then after email too. They wanted 50 seed of everything I had. Well, there are a lot of them that I'd like to have 50 seed of because they don't produce that many. But it's not an easy thing, and Dr. Sham Tollery he's doing a fantastic job, and I really appreciate his efforts, and I'm working, and we are working at it's a team effort here to get the material back into the national collection of materials that were once there. And about 5 years ago, I identified at least 200 accessions of wild peanuts. Well, since that time, it's more than double that, and we're still working on it to get the material back into the national collection so that he can distribute materials as he is able to increase them. He has minimum requirements that he needs to have before he can distribute them, of course. But we're trying to get as many materials back into his collection as possible.
Hey, thank you. Thanks for that lengthy explanation, because I think it gives us a sense of how much of a challenge it is to collect and maintain those collections once you identify the varieties. A question that somebody asked about one of the pictures. So in your slide deck, there's a picture of a lady that you mentioned was growing peanuts, one of the wild cultivars. The question is, was she growing it for her family's consumption, or was she actually growing it commercially so that she could sell it locally or regionally?
The wild peanut
Yes.
that they were growing for food?
Yes.
No, this was a tribe. I think there were about 48 or 49 people in that tribe, and they were growing that for their food supply.
Okay.
There was one. They took us to their slash and burn field. We got permission to go on to the reservation, and that took some doing because I was an outsider, and I had to convince them that I was an agronomist, not a person that's going to come home and make all kind of bad remarks about how they were treating the native people. But we got on to the reservation, and we're talking to them. There was a lady that was sitting in the front of her grass thatch hut, shelling what looked to be a wild peanut. And sure enough, we took a closer look. Dr. Viles looked at it. It was Arachis villosulacarpa. And so we inquired, okay, where are you growing this? And they agreed to take us to their slash and burn field. And then they showed us how they harvested, how they dug the seed out and replanted the plants in the field, and continued to perpetuate the species. Now, we've never collected that wild species in the wild, I mean, other than from the native people. And we would ask, where did this come from? And they would go into a long story about when they were on a reservation at another location, or give us a history of where it came from. And they showed us various things about how they planted them. But it was quite interesting.
Yeah. So another question came in. This is a really interesting question about the process that you've gone through. Can you describe the steps required to locate new wild species? Do you, as part of the process, do you start by talking to locals and showing them samples of peanuts so they can get acclimated with what you're looking for? Are there visual characteristics that you search for to help guide you on where to look?
Well, at the beginning, we were following information that Antonio Krapovicas obtained from old herbarium specimens. Like he would go to Paris, to the museum, and to Kew Gardens and other locations where and in Brazil, some of the materials in Brazil, to the herbarium, and there would be pieces and parts of wild peanuts that were collected by the Jesuit priests that went along on expeditions in the young explorers, in the early explorers. And there would be information like this peanut was collected 30 leagues south of Cuiabá. What's a league? In some cases, you look up the definition, it's 7 miles. In other places, you find 9 miles. So we go to those. They originally in 59, and then we followed through in later years, and after 76, we'd go to these places as best we could follow the information, and lo and behold, we would find them. Sometime we'd find a soybean field. Well, no peanut there, but it was interesting to see how accurate some of those things were once you figured out what the measurement was. But that was, it'd have been nice if they'd had a GPS back in when the people, when they first discovered the new world. It sure been helpful.
Yeah, clearly. So there's almost a part 2 question that someone asked, a follow-up question to that. So, Dr. Simpson, what places do you think are important to do new field trips to collect peanut germ plasma? What geographic frontiers do you see out there beyond the places that you've been to?
Well, Dr. Sejo has gone to one of the places that we were trying to get to in 1994, and we ran out of tar patches. We still had food, we still had water, but we had two tire patches left, and we were pumping up 3 tires every morning with a hand pump. So we decided we probably better get out of there while we could. But we were headed east in southeast Bolivia towards the Brazilian border. We were about 150 kilometers from the border, and we were finding different peanuts every time we stopped, just about. And I estimated that probably in the region where we were trying to get to, there are at least 10 more new species. Well, Dr. Sejo has been able to collaborate with the Bolivian personnel, and I think he's probably collected about 5 or 6 new species for sure, maybe a total of 10 in that area. So southeast Bolivia, northeast Bolivia is another place where there's still work need to be done. There's still collections to be made in Brazil. Dr. Valls goes out regularly. Well, the last year with the pandemic, no, but he still goes out and collects. But he goes out to collect grasses, but he also collects peanuts, and he goes out to collect peanuts, and he also collects grasses. But there are several places in Brazil that still need to be collected, and primarily northeast and east Bolivia would be my suggestion.
Yeah, sounds like lots of opportunity for future researchers as well to focus and build on what you had done. There's another question that came in, and someone asked you to talk a little bit more about stenosperma distribution, such as the difference between inland and coastal Brazil germplasm.
Okay. There's a trail of, and Dr. Viles is the one that I'm using his information here to talk about. He's the one that really ought to be telling this story. But there were people living on the west coast of Peru that went on hunting expeditions, and this wasn't a week long thing. This was a time consuming thing to go to the east coast of Brazil. And apparently along the way, they found Stenosperma, and they took it with them. They discovered that these seed were edible. They took them to the east coast of Brazil, and for many generations grew that species along the east coast and the littoral of Brazil. They grew that and made selections, and some of those seed are quite large. The pods are quite large on the ones that are growing along that coast. Now, when we first started this work, we could not figure out how those wild peanuts got washed there by running water or carried by birds, or how they got there. We were looking at, well, they washed down the Paraná River, the Paraguay, Paraná, down and got down in the ocean currents, went around, followed the ocean currents around the bottom of Uruguay, and were deposited on the southeast coast of Brazil. Then we got to looking at where the ocean currents were, and if it got into the ocean currents, it had to go to Africa first and then come back around to the southeast coast of Brazil. So that theory was shot to heck. And so then Dr. Viles did some reading about the Jesuit missions, and he got to looking at records of the Jesuit missions, and where the Jesuits were they were trying to convert the native people, local people, into their religion, and everywhere that one of those Jesuit missions was Stenosperma. And I didn't believe it at first when it was collected. Dr. Viles said, hey, this is Stenosperma. And I said, no, that's not Stenosperma. So I took some, brought some home, and I made crosses with what was originally Stenosperma, we thought. And the pollen stain was 99.9%. So it was obvious that it was Stenosperma. And we found that same peanut had been distributed well into the North Amazon, well into the west part of Brazil, to the southeast coast. But the original location of that species probably is somewhere around the central part of Brazil, Rondonópolis, in that area.
Okay. So we're running close on time, but I want to ask you one final question before we do wrap up. So based upon your experience in breeding and your research on the peanut plant in the past, what future peanut breeding advances are you most excited about, or do you see the greatest opportunity for in moving the industry forward?
Well, we're working with a new technology here that really has us excited. And this, I mentioned it in my talk, the Raman technology. And that with gene editing, I wish I was 40 years younger, I'll put it that way, to be able to learn and learn how to do this kind of work. But I'm not, so the younger folks are going to have to take it. But I think that's a real fruitful mission for the future.
Great. Well, I hope you have some comfort knowing that the work and the research you've done over years has built the platform for breeding that we'll be able to do in the future. Thank you for a very fascinating presentation, and more importantly, thanks for your dedication and a career in advancing the peanut breeding industry forward. And I'm very pleased to share with the group, because of your work and your research and your dedication in the industry, that the American Peanut Council will be awarding you the Lifetime Achievement Award later this month in New Orleans at the USA Peanut Congress. So congratulations on earning that award. It is very well deserved. And I look forward to being able to help present that to you in person.
I thank you. Thank you for this opportunity. I love talking about peanut germplasm and the wild species. It's just it's a lifetime for me.
It's very clear that the passion comes through. So thank you everyone for joining us today. This session has been recorded, and we will post it to the APC website with the link soon. And we look forward to seeing many of you at our USA Peanut Congress later this month in New Orleans, where again we'll be presenting Dr. Simpson with the Lifetime Achievement Award. Thanks everyone, and have a great day.